But there we go. We're here. Hey, Brian.
Hey,
We're back again.
We're live.
We're back again. Wow, we have a great discussion this time. So welcome everybody. And welcome Brian. We've got Brian Jamison with Diagnostic Biochips. We're going to be talking about finding your beachhead market, or what I like to call the market entry point. I'm going to let Brian introduce himself. I'm going to introduce him a little bit. So Brian's a client and a good friend.
So you better say nice things about me.
I better say nice. I always say nice things about you.
Look, I'm the client here, right? You pointed it out.
Oh, that's true. I can't trash you, but I wouldn't try. I wouldn't. I don't do. But I don't do that anyway. So I'm going to let Brian introduce himself and Diagnostic Biochips in a minute. But so we're going to talk about finding your beachhead market. I think it's a really important topic for everybody. For everybody launching, especially disruptive technologies. And Brian's got an extremely disruptive technology. That's one of the reasons I love working with him and his company. But also, I think it's underappreciated on how challenging it is to figure it out. And one of the things I love about this topic today with Brian is that we're in the middle of doing this. So Brian and I, we've been working on this for over a year now. So this is the conversation today is like a real live case study. And when I say live, we're in the middle of still figuring it out, right, Brian?
Yes, sir. The end of the story has not been written yet.
Exactly. We're somewhere in the middle. Hopefully we're getting close to figuring out the beachhead, but you don't know. But before we get into that, before we get into the discussion about where we are and what we've done so far and in figuring out the beachhead and where to start with for Diagnostic Biochips, why don't you just give us a little bit of background on you, Diagnostic Biochips, how you got here, and what you guys are doing?
Sure, happy to do that, and thanks for having me on. I tease you about being a client, but really it's been we're more like rangers going out there into the wilderness together on this one, and it's been great. Since sort of the day I was introduced to you, I felt like we were on the same wavelength in terms of really trying to figure out a focus for where we should be pointing this technology. And I'm an engineer founder. I'm a PhD engineer, and all of the good and the bad that comes with those things. It really makes, I think, a founder who's the technology person, it really makes you very obviously technology focused. Yep. And so I think it's probably a theme that's going to come up a lot today is, as a founder, getting help in the areas where you need help and not being shy about that.
Yeah, I must say though, you have taken on and embraced the market development side of things and the non-scientific aspects of it really well. So, but tell us a little bit about your basic technology and what
Sure.
what your vision is. And
happy to, yeah. And I'm going to start. Maybe I'm going to annoy you with what a high level I'm going to start at. But let's start with Chris, and let's start with what makes Chris Chris.
Okay.
Do you know what makes Chris Chris?
Good question. I think I figured out some of the psychological aspects of it, but I'm not really so sure maybe we're talking about that.
So really at a fundamental level, what makes Chris Chris is the Chris network.
Aren't I supposed to be interviewing you, by the way, here?
Tough luck. That's what you said. It was going to be brutally honest. The teaser for this, I think, said brutally honest. So here we go. I'm being brutal. Okay. Yeah, there's a Chris network up there. It's called your brain.
Yes.
And this is all the really important stuff to me, and I think probably anybody listening would probably agree, all the really important stuff about, you being you, your memories and your ability to, sort of execute motions and plans and recognize people and have conversations and feel emotion and learn and all of that stuff.
Right.
It's a network property. There's no one neuron in your brain that makes Chris Chris. And it's probably so obvious to everyone that it almost doesn't even bear talking about. Yet, when you look at sort of the history of science and the history of neuroscience and the brain, there's been this incredible blind spot around networks. we act as if, and I say we, I mean broadly and generally, with a huge number of exceptions, but the field has acted as if we get really, really smart about how one neuron works, or one channel on one neuron, or one, biochemical pathway, we're going to suddenly understand how the brain works.
Yes.
And there's a little bit of first things first. Those things are important. But also, there's a little bit of just, poor ability to engage new tools to ask those really important questions about the network. And to some extent, that's been a technological limitation. We have not had great tools. I was very, very lucky. I looked for this question, like almost as a child, teenager, child, like, how does this brain network work?
Really?
And I was lucky enough, after my undergraduate, I was a physics major, to find out about this guy named Ken Wise at the University of Michigan, who was really pioneering the idea that we can use semiconductor processing fabrication techniques to develop minimally invasive, very capable sensors to interface with the brain.
Got it.
So the rest is history. I went to graduate school and got a PhD with Dr. Wise specifically to use those techniques and apply that to neuroscience. And the rest has been history. I've kind of had some detours. I went to work for NASA, strangely, for 6 years. I say strangely, it was an amazing experience. And it turns out I learned about instrument development at NASA. So that has become very important, in my life now.
Sure.
But all of our basic technology is predicated on semiconductor, high density, high performing chips that can be implanted in neural tissue and give a real time readout of network properties.
Got it.
Yep.
Interesting. Well, I mean,
I've never told you this before. How?
I don't know. I didn't know. I've known parts of the story, but not. But the stuff about you as a kid being curious about how the brain operates, and the other thing we haven't talked about is this network piece of it. Is that much? And that's kind of what drives all the underlying things that you're doing, is understanding how the network of neurons work as opposed to individual neurons.
Exactly. Yep. And I'm not trying to oversell. I'm not saying that single cell behavior, neurophysiology, neuroanatomy, that these things aren't important. I'm just saying it's not sufficient to understand the brain.
Yeah, and it's interesting. You and I have talked about this before. I mean, the complexity. I mean, I've worked in genomics, and the complexity of the genomics and biology, and extremely complex. But even that world's much more known today than what's going on inside the brain. Is that an accurate statement?
I think it's fair, but you have to turn back time to, whenever it was 1952, I think, or, roughly when, the structure of DNA was discovered, and, realized that all of that learning in genetics has happened, more or less in our lifetime. I wasn't born in 52 quite yet, but, in our lifetimes, that whole thing has unfolded. So if we can drive a similar unfolding in neuroscience and in the therapies that should come from that improved understanding, in the lifetime of our kids, for example, over the next 50 years, that's going to have a huge impact on real things, like disease, neurodegenerative disease and neurodevelopmental disease, et cetera.
And if we look at the aging population, what's going on? I mean, it's a huge issue today. I mean, Alzheimer's and just dementia in general.
Yep.
And as we live older and older, which some of that's slowing down a little bit, but as we live older and older, this becomes more of an issue for us, both as a societal thing, but also as individuals. So let's shift. I mean, so thanks for sharing. And tell us what Diagnostic Biochips is doing today. Just give us a little bit of that.
So your background. We're at this really important inflection point, and it's we've been at this for a while. We founded the company in 2013, and there's sort of a before this moment and a now and everything after. Before this moment, we've been very, you talk about a beachhead market. Really, our beachhead market, going back for the last 10 years, has been the very, very basic science research area.
Yes.
We have sold these sensors, these high-performing, high-density sensors that have been used exclusively in vivo. So we're going to talk today in vivo versus in vitro, just very simple distinction. In vivo just means in life, literally. So in a body. So these are, usually animal experiments, rodents, non-human primates, sometimes humans.
But real basic academic researchers, basic core research labs is where you've been.
Not not, drug discovery, not I'm trying to find a therapy, I'm trying to find a treatment, a new drug. It's just like, we talk a lot about the hippocampus as one particular structure, very important structure in the brain. Stereotypical customer from that era of the company, I want to understand how the hippocampus works, how it codes information, and how it leads to the storage and retrieval of memories.
Right.
That was like our ground zero, has been and will continue to be in that market, a ground zero customer.
Got
Basic
it.
science space.
And that kind of brings us up to when you and I met a little, probably a year and a half ago or so. We were introduced by a friend of both yours, Rick Satcher. And when you and I first started talking, what you asked me to help you do was sell to pharma clients, right? And I think at the time you had one big client at the time. And so tell me, so tell me, what did we discover in the? So we started, and I remember the first thing I told you was, let's go do some customer discovery. You have one client. Let's go figure out. And use customer discovery to figure out where the market opportunity is in the rest of pharma.
And we've probably talked to close to 100, maybe not quite 100 labs, CROs, pharma, researchers.
In a short period of time, we covered it all, right?
Yep.
So, and the task was, let's go get some more pharma customers. Kind of talk us through that. What did we learn? What came out of that?
So I think we have to finish the story we talked about before. So what made us think suddenly, 18 months ago, 12 months ago, maybe 2 years ago, that we could make this pivot successfully into what we have now come to call, translational markets, meaning markets that aren't basic research, but they're more oriented towards drug discovery and therapy development. So what happened? Honestly, we have to talk about stem cells and induced pluripotent stem cells, iPSCs, and we have to talk about organoids.
Sure.
Because that really is the technological development that we had nothing to do with.
Yep.
Suddenly seemed to us, who have been out this for a while, to come out of nowhere and start going gangbusters. So these are stem cell neuronal cultures that start to oversimplify, that start to look like mini brains. And what we saw in the market, we saw all these researchers and people mostly at least aligning with drug development, who were developing these beautiful disease models using organoids, brain organoids. So when I say a disease model, I mean these are human stem cell cultures, so they behave like human cells. And they were able to genetically introduce modifications that made an Alzheimer's-like brain or a Parkinson's-like brain or an MS-like brain.
Right.
And what they were lacking was a readout. So you have this biological model. What do you do with it? We can slice it and stain it and look at anatomical features and, fluorescent mapping of different channel types and, expression of different genes and all these things. But from, again, getting back to our bailiwick, which is dynamic systems and how networks operate.
Yep.
Now you've got this great system for asking at high throughput,
right?
Because you can put a bunch of these on a little 96-well plate.
Right.
You've got this great system for asking wonderful network questions at scale.
Yep.
What questions are we asking? We finally have tools, and to our eyes, it looked like not the right ones.
Right. The ones that we currently had. Yeah, but the thing that was interesting from diagnostic biochip size is you had kind of a convergence of the biological tools and the tools that you've been working on for all these years. There was an opportunity here. So tell us what was going on with the big pharma customer and what we found out early in the discovery, going beyond that one customer.
We, you talk about customer pain points, but we as a company had a pain point, which is we had this terrific, technology. And the fact of the matter is the way we were using it and working with customers and clients to deploy it was inherently low throughput. It did a surgery on an animal.
Yep.
And now suddenly we have this model that's at scale, and there's this really strong pull from pharma about let's figure out how to deploy this. God knows we need better preclinical models for drug discovery. Yep. This feels like one. How do we do it? And you mentioned, I'm not going to say the client by name, but a pharma company that everybody here has heard of that was very, frankly, very forward thinking and saw the ability, saw our technology and its in its then form, and saw how it would fit into this, high scale organoid model, and said, let's go, like, let's do a pilot project,
Yep.
let's figure this out, let's use your technology and do a couple of case studies with different disease models. And that was our initial traction. That's when you and I met, and it felt like fantastic. We're off to the races.
Yep.
All we have to do is find 5 more customers like that one,
Yep.
and we have a real business here.
Yep.
That's what it felt like at the moment.
And it was a pretty big contract. It's in the hundreds of thousands. It was less than $1 million, but in the hundreds of thousands of dollars a contract.
The investment to date has been actually north of $1 million. So for a small company like us, it's been made a huge difference.
Yep. And okay, so and we came in and you said to me, let's start selling to these. Let's go get more of these.
Get more of those.
And so that's what we. But I said, step back for a second. We've got one. Let's, instead of going into sales mode, let's go into discovery mode.
Yes.
And figure out where those customers are and where the pull from more of those type of customers are.
And I do have to say, in retrospect, maybe we weren't quite as naive as it makes it sound like we were, because we knew that we didn't have a commercial instrument at that point. We had a prototype.
Yep.
And a mutual friend of ours who's probably on the call right now calls it a peach basket.
Yeah.
This ugly, crazy looking thing that if the client is friendly, you're happy to put it in their lab and work with them on it. And, every once in a while it throws up a red flag and says, I'm in trouble. And someone has to pull the chain and fix it. And that's what we were.
But you also had the customers willing to pay you pretty big bucks to start playing with that prototype. So.
Exactly. We had a very sort of solid notion that we had to make this easy. That everybody wasn't going to be as patient.
It was too garage based at that point in time. We had to simplify, and we're working on that.
So we went out. Yeah, just to remind you, we went out and we figured, hey, this is easy. We can find, the person at the one company that, our path in and how we connected and how we got in there. We can just recreate that and do sort of the same thing and have similar conversations, and everybody's going to love this, and we're going to be off to the races. And so that's where we started, our focus on pharma and obviously the CROs, the contract research organizations that serve pharma also.
Yep.
And to be fair to us, and you and the team here, we did have those conversations.
Yes, we did.
With pharma and CROs, and there's nothing that we've learned, nothing that we have heard has dampened our enthusiasm to believe that that's the perfect long-term market for us.
Correct. Yeah, which is good. However, what we did find was, did we find that they were ready for us today?
No. No, and so and really through no fault of our own. I mean, I think that the whole sort of wrap on organoids and iPSCs in pharma is great technology. You can pull up quotes from Roche, from Hans Cleaver, who's the VP of R&D at Roche, really saying this is the future. Organoids are the future. But the actual people on the bench are all scratching their heads trying to figure out exactly what to do with it, with the base technology. And importantly, they want to have proof points.
Yeah.
And the place that they are looking specifically for proof points is peer-reviewed literature, journal papers, conferences, and their colleagues that are more academic, not academic like the basic science customers.
Correct.
But the academicians who are involved in research that directly reads on drug discovery.
Yeah, it's interesting. So I think part of this is the mindset between a pure academic researcher and what a commercial industrial client is looking for in an early technology.
Yep.
The researcher is willing to do the basic research and answer those really tough research-based questions. Someone in a pharma, it's very unusual to get someone that's really to look at the technology. They're more asking the question, not only does the technology work and does it start to address some of the things they want, but what are the specific use cases going to have a direct impact on moving my business ahead from from a functional standpoint, but also from an economic standpoint.
Yep.
I think that's what it was. So I think, and this happens all the time with startup companies, is they get one client and think they have a market. That's one of the reasons I said, let's do the work and do the discovery work and see where customer number 2, 3, 4 is 5, and not try to sell them, but let's do the discovery work. So as we're having those discussions, if we don't get sales, we're learning. But if we do get sales, we're learning too. So we're learning either way. So it's a win-win, no matter what the outcome was. Is that a fair description of what we did?
Yes, 100%. Agreed. Yeah, we never, all through those discovery calls, we never sort of went to close, put the screws to anyone, tried to get them to place an order, even told them. In fact, we led with, we're not selling anything today.
Yes.
Right. And always something I had a hard time with that you had to coach me a lot on is like, stop talking.
Yeah.
Try to, we talk a third, they talk 2/3.
Yes.
And just get them talking about their pain points and see where that lies.
But here's another thing I think. So did we lose any of those contacts because we didn't have sales conversations with them?
Am I giving you the wrong answer? I don't think we did.
No, I think that's the answer I was looking for, but I just said, so there's not a downside to approaching these potential customers with the discovery mindset early on. There is a downside to approaching them with a sales mindset early on because you limit your ability to learn from those conversations.
Yep. It is hard though. Like I remember feeling very awkward. I mean, look, let's face it, by definition, the people that we're reaching out to are heavy hitters.
Yes.
Right. They're thought leaders. These are impressive people that run labs, whether they're, tenured professors at fancy universities, whether they're working at some pharma company, group leader, and they're very busy and they tend to be very to the point and, punchy, and they want to hear it. And now you're calling up, fear was now you're calling up and you're like, hey, we don't really have an agenda. We're just here to get you talking. I mean, obviously you don't say that, but
you'll say
that's
that.
the vibe that I always feared that would annoy people.
Did it?
No, it generally didn't. I can think of 1 or 2 people, maybe it tipped over into that a little, and we were smart enough to pivot, and you give them some meat and potatoes about our technology and get them hooked and interested. But generally, no, people love to talk about their research and their problems and their pain points.
I think that's I've had that experience myself personally. I know a lot of people have that experience, particularly challenging for a CEO of a company too. I think and I think you did a great job doing it, and you took the coaching well. So what did we learn?
Yeah, I was going to say, let's cut to the chase. So we ended up having to coin a term, and I think everybody's going to know what we mean. Yeah. We coined this term translational labs.
Yes.
And the reason we coined them, that we sort of started thinking about things that way, was they weren't that basic science, remember, in vivo sensor world that we came from, the basic, like, how does the hippocampus work? But they also weren't full up, like, we've got this Alzheimer's drug and we're trying to bring it to market. They were something in between that. So they were people that a lot of them are focused on organoids, creating and validating organoids as disease models.
Yep. Yep.
Some of them are actual suppliers who are trying to supply organoids to the chain, and they want to know, we've got a metric, a quality metric for knowing we've got good organoids. Some of them were actually doing drug discovery, but in more of an academic setting. Yep. Like they've got one compound they're trying to test in various forms, and they want to see how this impacts, a model like a brain organoid. So that became our sweet spot, and I would say we started getting like 5 respondees that looked like that to every, or maybe 10 to every one call in pharma that we could take. And the appetite was much higher. They were like, here's our pain point. Yes, we need a better readout. We're not happy with what we're doing now to read these beautiful models out. And what you guys have feels like it would be the right thing. Like, convince us, show us data.
Right. Yep. Right. They actually had, whereas the big pharma companies, their problems were too far removed from where the technology was, and frankly, the organoid research was, to be able to say, here's our problem now that we need to solve to help us move our our programs forward. Whereas in these translational, what we call these translational research, some of them were academic based and a lot of them were not. But they actually, because they were kind of that quasi research, quasi translational into the industry side, drug development side, the problems they were having today, they were working on were problems that they wanted to solve today.
Yes. Yep.
So tell us talk a little bit more about that. Give us a flavor of that. What type of conversations did we have with them? How did our discovery conversations progress into?
Yeah.
Because it's been messy. It hasn't been clean.
True. It's been sort of 2 steps forward, 1 step back, and a little squiggle. But it did happen very quickly. When you realize I think our first discovery call was like just about a year ago, maybe even less. And now we have, we've got POs, pre-orders on our instrument. But in terms of early access to our prototype instrument, we've got an instrument out, not in that one pharma lab we talked about only, but in just under 10 translational labs all over the world.
Right.
And that happened within, we did an install last week, so they're still ticking along. But that happened like that because that initial discovery call kind of became, follow up with data for us to know that this is real, which we had ready to go. And then once they saw that data, they were like, man, how do we try that? Well, not everybody, but thought leaders, the people that are our Beachhead type customers. Let's talk about this. How do we get data like that?
And describe that customer. What's that early customer look like? You said thought leaders, but is there a characteristic to them that you're learning?
Yeah, it sounds like a silly answer because it kind of they define themselves.
Yeah.
Their appetite for moving forward and for wanting to try a new technique under circumstances that were very clear. This is not a full commercial product yet.
Yeah.
That defines them as a thought leader. But there were times, like there were questions that we would get on these discovery calls where we knew like, oh, this person is or isn't that kind of person.
They would disqualify themselves as a thought leader too.
Not disqualify them forever from our interest in pipeline, but sort of knowing that this is not a lab where we should make an investment to go in and get early data.
And this is a really important. There's 2 components to this that are really important in finding your beachhead. One is this, by using this discovery way of and asking questions, and then also pushing hard into it with something. The product we're still pitching is this prototype, which is basically build in the garage type of product, and so part of it is this disqualifying who the innovators and early adopters are. One of the things you guys found with the early pharma client was a really early innovator. And those people they're even different than the early adopters who are visionary to take risks, but they're willing to innovators are willing to take on the technology long before it's ready and kind of what we said, play with it and figure it out. The early adopters, they're more driven to solve a particular problem, which is what we were discovering with discovery, but they also have this visionary mindset. So they're not, the early majority, and I've talked about this, they're driving to solve a problem, but they're not a risk taker visionary. They want the problem, they want the solution to already fit the solution to the problem, have all that evidence. The early adopters, they're solving problems, so they're very pragmatic, but they're this visionary. So they're willing to say, okay, listen, I want to start bring the future to me instead of waiting for the future to be there when I'm already there. So it's really important in these discussions. One is that. The other thing is that people will self-select in and out of that, which I'm glad you brought that up because this is really interesting. So tell me, all of these 10 sites, so we've got this one big pharma client. The rest of these 9 or 10 sites are these translational group, which is pretty small community. It's not a big market. There's hundreds of thousands of research labs. There's probably thousands of pharma companies we could sell to, but this is probably a pretty small group. Give me pilots and how we selected people and how that shifted over the last 3, 4, 5, 6 months as we progressed along. Talk to us a little bit about that.
So I guess there's a couple questions in there. I think one is like size and timing of that intermediate market, translational. Like
Yeah.
how do we? It's not a huge market. It's not a massive market in the way that preclinical tools for CNS drug discovery is a massive market.
Yes.
It's a non-trivial market, but it sort of still feels like a road to something else. So how do we grow and enter there and join it, and not have that be the last thing and never sort of make it across that chasm? So this is something, that bridging the chasm, right?
Yep.
They are still on our side of the chasm, right? The
Yes.
chasm is really in between the translational and pharma, and we've got to sort of get our acts together here, roll out a full commercial instrument, create the market momentum, and that's our beachhead market,
Yep.
translational labs.
Yeah,
And,
yes, absolutely. But the important thing you point out here is that it's not an isolated market in and of itself. It is connected with the larger pharma market that we want to get to, and it's a precursor for it.
yes. So here's a really interesting stat. Like if you start poking around in lab head type, entry level, but PhD entry level job openings in preclinical drug discovery,
Yes.
and zero in specifically on CNS. So
Yep.
most of the major pharma players now, there was a lag for a little while, but most of them have a pretty healthy CNS pipeline. It's second or third biggest disease area now.
Yep.
And you start looking at job ads, there's a lot of activity around recent PhDs that know how to culture brain organoids.
Oh, wow.
And so what my point is that it's clearly like there's a porous division between these translational and academic labs and
Yep.
pharma, and our belief and experience, and we can see this.
Yes.
So this. It wasn't how we got in the door, but our
Nope.
biggest champion at the big pharma client came from a lab where we're doing, like, he went over to this company
Yep.
from one of our pilot labs. Now, he made that jump before we started working. We actually swam the other direction from him back to the
Right.
translational lab. But my point is, there's a bridge there, and part of our plan, what we have to do is sort of go with this generation of young scientists that's going to be taking this technology over to pharma and teaching pharma how to build these models and do the readouts with our.
Yeah, there's lots of ways to bridge this, and this is this crossing the chasm. It's not as if you plan for it and you design your company to build it to cross it, it's not as deep and as wide as they make it out to be. And you and I haven't talked about this conversation. It really isn't. It's still really challenging, as you and I have talked about. One last thing I want to leave people with a little bit before we wrap up here, Brian. This has been I love this because you and I are talking about, really how we're doing this not over the last year and still doing it today. Tell us paid pilots versus non-paid pilots and how we're navigating that early on. It's really interesting. The biggest mistake most companies do is they don't ask people to pay them for their pilots. That is the biggest mistake I see. So tell us we're doing.
I'm glad, first of all, I kind of knew this question was coming because I've been thinking about it nonstop for the last 24 hours. And we've been pretty loosey-goosey, I have to say. Everybody participating in our EAP, Early Access Program, everybody has paid.
Yes.
There's a however coming. However,
yes.
Some people have paid in the form of real and significant sweat equity in the form of proposals. So we have put in proposals to the state of Maryland, with an adapter lab where we took pilot data, we worked together, we put the proposal in, and we were funded. Significant significant funding. Another early adopter lab, we put in an NIH proposal, a non-trivial multi-year NIH proposal. Yep. And she, that PI, Flora Baccarino at Yale, put a lot of time and effort and heart into that proposal.
Absolutely.
More than stroking a check for some hardware. Other people, and we support everybody, of course. We want We want very happy customers at this point, delighted customers. But other people have stroked a check. We've gotten a PO. The way we're handling it is, and we just kind of made this number up, half the purchase, the final commercial instrument purchase price, which we are rolling out in November. So we're already taking orders. So that's no longer this Frankenstein peach basket. It's a legit, straight up, self-contained, beautiful instrument.
Yep.
And their half, the early action people, their half purchase counts 100% towards that instrument purchase.
Yep.
And we have had people that paid that way. So yeah,
I just want to point out for everybody, that's not an insignificant amount. The price of the product's $150,000, so that's a $75,000 check being cut to diagnostic biochips for that. So
we have plan participants that have gone that route. We have others that have done the proposal, et cetera. So but it's been dynamic. I mean, believe me, because
the really important thing is everybody's paid in some way, and over 3 or 4 of them have paid it with writing a check out for it. Yep. And the other thing I need to point out for everybody, this is a product that's still the prototype. They're paying for the prototype today. That's really important, because this is, and you and I have talked about this. It's not so much the money. The money is always important to a small company, always. But it's important that it validates that market pull that we're tapping into is actually people are willing to pay, pull out their checkbooks and pay for that as well.
Yeah, there's really 3 reasons here. You hit on, I think, 2 of them. One is anybody that's rolling out a significant new product, by definition, you're probably, in a tricky place with cash.
Yep.
It's an expensive proposition from development and marketing and support and all that stuff. And we're trying to be very, very efficient with our capital. So we need the money, frankly.
Yep.
2, it's market validation. So yeah, someone can say, sure, come up to our lab and install this. It's no skin off my back. I'm not putting anything into it. What do I have to lose? Yep. So they may participate, but not really believe. And the money sort of validates their seriousness. But the third thing is external optics. So, whether it's with funding sources, I mentioned the NIH, the state of Maryland, TEDCO, or investors, people want to see your pipeline, and they want to see that you have some amount of commercial traction. And it makes a huge difference to be able to say, as we have this EAP, it has generated this much cash. We have an actual instrument pipeline on top of that, X number of pre-orders teed up already, ready to go.
And just to be clear for everybody here, we're still 4 to 6 months from having the commercial product ready to deliver. And we're talking with people about pre-orders of that, right? And we have some pretty aggressive sales metrics between now and the end of the year on that product that they won't be able to take possession of until probably late first quarter, early second quarter.
Let's say mid quarter 1. Raymond.
Okay, mid quarter 1. Okay, but still, we're still 4 or 5 months away.
Yes.
So, Brian, this has been a great discussion. Any last thoughts or any recommendations around this topic you would have, or any last thoughts you want to leave with other founders?
No, I mean, first of all, I want to thank you. I mean, it's been great having you in our corner, and you're not alone. We've got a product launch and marketing team that has been awesome. Grace and Bruce and others, Building 12, that have helped. And it's just, again, I would emphasize, you're kind about my skill set, but I'm an engineer, and it's it really takes a team to launch a product like this and understand the market. So thank all of you guys.
Well, it's been my pleasure, and I look forward to continuing on this journey. It's been fun so far, really hard, but fun. But it's going to be fun to see how this continues to play out over the next 1, 2, 5, 10 years. And I think the most exciting thing, Brian, is getting back to where we started the discussion, is ultimately the clinical impact, and the impact on neuroscience and the drug discovery, but most importantly, how we can eventually impact humans and patients with what we're doing today. So,
yep.
Brian, thanks. It's been a pleasure. Stay on. We're going to sign off, but stay on for a couple minutes and we'll wrap. So, Laura, you want to wrap us up? So thanks. And everybody, we'll be back again soon.